Queenie. I'm a professor and vice chair in the department. Bill Hirsch, our chair, is away, so it's my pleasure to introduce Christina Turner today. This is her symposium, and just as a reminder, or for those who are not in the department who are watching this either live or later when it's recorded and posted, the symposium is part of the process for our dissertation students, in which they're really thinking about the background, kind of the prior art, where the real, kind of open questions that they can address. And so this is that synthesis of the literature review and thinking that they've been doing in terms of the ideas that will really be the kind of formation for their proposal. So they do the symposium, then they'll actually develop a proposal with their DAC, and then they'll actually have a proposal defense that will again be live. So let me just tell you a little bit about our speaker today. So Christina is one of our NLM pre-doctoral fellows. She did her Bachelor's of Science at Occidental College in 2014, and also did her master's at Western Washington University. Her background is in molecular biology and microbial ecology. And she mentioned, and she actually wrote her own introduction, which I'm paraphrasing, but that she loves to think about microbes and their interaction with the environment. And I think you'll see a lot of that today. And she's also fascinated about thinking about the human body as a complex ecosystem. And you will definitely see some of that thinking today. And she really would like to understand the environmental impact of microbial interactions and host health, viewing health as a holistic approach to improve someone's physical and mental well-being. She is from Portland, and she looks forward to seeing where this project takes her. And with that, I will stop talking and hand it over to Christina.
All right, thanks, Shannon. Yeah, I'm excited to share some ideas I have and what I've learned so far about the gut microbiome resilience as a metric to predict overall human health. And I mean, Shannon kind of already got to this point, but this topic or idea is pretty much based on my interest in applying our understanding of the gut microbiome to better understand our overall health and well-being. I'm going to start, oops, oh, how do you change? Okay, there we go. I'm going to start by sharing the overarching question that kind of motivates this, and then some background information on salutogenesis, gut microbiome and disease, and applying this concept of ecological resilience. I'll share our problem space, or what we know and what needs to be done, some prior art and current approaches, and then I'll share what current gaps are still there and potential opportunities. So the overarching question that motivates this topic area is kind of represented by this diagram I made of twin A and twin B, representing two individuals who have the same sex, very similar genetics, diet, lifestyle, what they do, any medication or comorbidities they have. Let's say all the variables are very, very similar to the same, and they're exposed to the same disturbance or challenge. So some examples would be like a loss of a loved one, a car crash, a heart attack, or stroke or cancer. What's interesting though is that even though all the variables are the same or similar, twin A might come out of this disturbance with very poor health or no recovery, versus twin B has a quick recovery back to the original healthy state, or maybe the disturbance doesn't affect that person's health at all. So then the question comes up, why can two very similar individuals who are exposed to the same disturbance respond so differently? And rather than trying to identify all the factors involved and characterizing all the variables, could we predict this overall health response by defining and developing a metric? To best answer this or tackle this question, we propose to take a salutogenic approach. And this salutogenic model of health relies on this concept of understanding and viewing health as a process along a continuum between health and total ill health, so ease and disease. And this is different from this traditional pathogenesis approach that kind of looks at origin of disease or how diseases are developed. So it's a more holistic approach to understanding health. And salutogenesis really views the human body as a system that contains subsystems. A subsystem of the human body is the gut microbiome. And it's well known that the health of the gut microbiome is really important in maintaining the health of the host, which is us. So gut microbiome, or more formally, the gastrointestinal tract microbiome, I probably will be referring to this as gut microbiome, or GM, just as a note. The microbiome is a collection of microorganisms that include fungi, archaea, viruses, and bacteria. And these microbes are closely involved with the host, which is us, and they have intricate interactions to maintain this healthy ecosystem. And as a note, the microbiome is composed of all the microbes, but I'm going to be mainly focusing on bacteria because that's where a lot of the research is. So this left diagram, you can see the healthy gut ecosystem with different types of bacteria. So it's very diverse. There's a lot of different shapes and colors of bacteria. And in this healthy gut, the gut barrier is maintained. So you can see the intestinal epithelial cells are very tight knit, and they don't allow anything in the luminal space of the intestine to come through into the bloodstream. So this is the gut barrier, and a healthy gut ecosystem helps maintain this. When there's a disturbance or challenge, whether it be internal or external, this community of microbes can get disrupted, and the balance of what microbial and at what abundance can be disrupted. And when that happens, it can go into this dysbiotic state. Commonly, this dysbiotic state is characterized by an overgrowth of one or two types of bacteria. And when this happens, this ecosystem, the normal processes are dysregulated, and this gut barrier can be impaired. And when this happens, microbes and microbial products can get past this barrier and then into our bloodstream, causing endotoxemia. And endotoxemia often leads to inflammation. So in this situation, this is usually referred to as gut microbiome dysbiosis, and it's associated with many diseases, including inflammatory bowel disease, colorectal cancer, obesity, and type 2 diabetes, as well as liver diseases and also Alzheimer's disease. So it's well known that gut microbiome health is really important supporting our health, human health. And given that the gut microbiome is a subsystem of the host, the human, it's crucial to understand how the gut microbiome health works. One of the proposed metrics to understand gut microbiome health is this concept of resilience. And so I'm gonna, whoops, sorry. Okay, there we go. So I'm gonna kind of talk about what resilience is. And I'm using the definition of ecological resilience. And generally, it's defined as the ability of a system to resist or recover from a challenge. And it is important to note that there are many different definitions of resilience, but I'm going to just go with this general one. And if you look at this panel down here, this represents a stability landscape. Also can be called a ball and basin diagram. The ball represents the system. So let's say it's the gut microbiome for this instance. And these basins or divots represent a state. And if a disturbance occurs on this system, so here this shows this external challenge occurring to this individual, which then affects the gut microbiome. And this gut microbiome is at this tipping point or tipping critical threshold between states. So from here, if the system is resilient enough, it can resist that disturbance and return back to its original healthy state. Versus if it's not resilient enough, or if the perturbation or disturbance is frequent and strong enough, the system can be pushed into this alternate stable state. The clinical implication with this is that this alternate stable state can be resilient, and it can confer a negative phenotype to the host, which this process occurring with the resilient stable state with the negative host phenotype has been proposed to be linked with chronic diseases. So diseases that treatments don't work or have a hard time reverting to that healthy state. So there are some characteristics of microbial communities that have been found to kind of underlie resilience of the gut microbiome. And these are kind of key characteristics. So I'm going to quickly explain them. Microbial diversity and functional redundancy are kind of key. So diversity, simply put, is just the number of different kinds of microorganisms. And then the functional redundancy is this phenomenon that microbes do where you can have phylogenetically distinct taxa that can play similar functional roles. So this diagram on the left-hand side shows the distribution of the types of bacteria. This panel shows for oral microbiome, so your microbes in your mouth. And it shows that firmicutes, the green one, is the predominant bacteria. Compared to the gut, which looks very different, it's predominantly Bacteroides. What's interesting though, when you look at the functions of the microbes, so what they're doing, not just who's there, the distribution of the functions based on the colors, it looks very similar. So this diagram is trying to show the idea that even if you have different taxa in your communities, different types of bacteria, you can still have similar functions. And functional redundancy and the diversity is important for resilience and in gut microbiome for keeping resilience because, as an example, if you lose one type of bacteria that confers an important function, if you have a functionally redundant taxa, they can kind of replace that lost function and keep the microbiome doing its normal healthy functions. Some methods that are used to characterize the microbiome are 16S RNA targeted gene sequencing and shotgun metagenomic sequencing. And I want to briefly go over these because the approaches I'm going to share mostly use these. And there are different levels of methods used for microbiome. So I'm focusing on methods that use DNA as an input. But just so everybody's aware, there are methods that use RNA and protein and metabolites. But a lot of the approaches are not there yet. So you get DNA from host samples like skin, feces, or saliva. You could do this more targeted approach that characterizes just bacteria. And you can look at the phylogenetic diversity, so different types of bacteria, and also the abundance of taxa, or types of bacteria. And then the shotgun metagenomic approach captures all the genes, including host genes. And with that, you could get microbial gene counts, and then also kind of predict what kind of functions are occurring. From this data, you could do analyses such as alpha and beta diversity analyses to compare differences in taxa within samples and then across samples. You can also do statistical and machine learning approaches to predict the distribution of data. You could also do co-occurrence network analyses to understand what microbes are present and how they're interacting. A lot of times you do this for a pre and post disturbance to see how the interactions change. You could do something similar, but with metabolism to see how the microbial functions change and interact with each other. So that's kind of the background, and I'm going to get into the problem space. So what we're dealing with. What do we know and what do we still need? So we know so far that the gut microbiome plays a significant role in our health. It's agreed that defining what a healthy gut microbiome is nearly impossible, and this is because of the inter-individual variance in our microbiomes across individuals. The gut microbiome is also a very dynamic and complex community. So characterizing the changes and defining one healthy definition is tricky. The importance of this idea of resilience to maintain a healthy gut has been found, and it's agreed upon that resilience is key. And then defining what a healthy human is also kind of challenging. It's been proposed that using this gut microbiome resilience as a surrogate for human health, and this is just to note, this is just a proposal. There's no data or any validated models to support this yet. It's also been theorized that this gut microbiome resilience and human health have a relationship, but there's few data to support this still. So our overarching need is we need a metric for overall human health, and gut microbiome resilience has been hypothesized to be a promising candidate. To further kind of assess that question, we need a robust assessment of gut microbiome resilience metrics, and then ultimately we would need a validated model to quantify this resilience and then apply it as a potential indicator of overall host health. So I'm going to share current approaches that apply this ecological resilience to understand system health. And because resilience has been applied to a wide range of systems and in very different ways, I organize them into 3 categories. So one of them, they apply resilience to measure, or they apply metrics in ecological models to measure that resilience in systems like coral reef or soil systems or rivers. And then they also apply the idea of resilience to measure human health. And then the last category, they apply resilience to measure the health of the microbiome. So I'm going to kind of go through each category and talk about the approaches. Okay, so the first few approaches in this environmental systems area, Scheffer et al. Proposed using this concept or phenomenon called critical slowing down to measure dynamic indicators of resilience. And this phenomenon, which I find really interesting, called critical slowing down, it's an early warning indicator of a tipping point. So if we just use this as a reference, here's an attraction of basin in a system. And the idea is that when the system gets closer to this tipping point, the recovery rate slows down. So to estimate if a system is going to be pushed into another state, you can measure this critical slowing down to predict that shift. So this approach applies that critical slowing down, and the key assumption with this approach is that they assume that the system is exposed to many small perturbations rather than like one acute disturbance. The advantage of using these time series data that kind of captures these small perturbations, you can estimate resilience based on those to calculate the pattern of micro recoveries. So micro recovery would be how a system kind of recovers back to that original healthy equilibria. And then based on this framework of critical slowing down, they were able to identify measures that characterize it, and they termed them these dynamic indicators of resilience, or DIORs. One of them is variance. One is variance, which can be seen here with temporal autocorrelation. So if you look, this left panel represents a system with high resilience, and then this right one represents a system with low resilience. And the idea here is that a system with low resilience has a slow recovery back to that initial state shown here. And then you can estimate these fluctuations that occur along the time series. And based on those fluctuations, you estimate these measures. In a state that has low resilience, you have higher temporal autocorrelation and variance seen with these crazy jagged lines. And this represents the correlation between the states and the changes in the states. The next characteristic that indicates resilience is cross-correlation. So that's correlation in the changes between different elements of the system. And high cross-correlation indicates a lower resilience state, so that's represented here. And this approach is still a proposal. It actually hasn't, they didn't test it on any model, so that's probably important to note. And they also propose that you can estimate these DOIRs from vital parameters. So some of the examples they give is like blood pressure to predict somebody's frailty. Another parameter they propose is a mood for things like risks of depression. And then specific for the microbiome, another parameter they propose is like the intake, the amount of fiber intake, because fiber is associated with a stable and healthy gut microbiome. Another approach applies the concept of entropy and data complexity to assess resilience of a system. They measure the change in this entropy measure they use called singular value decomposition. And they calculate this from a multivariate biological metric. And this workflow kind of shows you start with this multivariate data set, you use this SVD method to calculate entropy, which is then associated with complexity of your system. And their approach then takes this measure of entropy and exposes the system to a disturbance and compares the change in complexity and entropy. And if you have a higher complexity change, then they estimate that that system is resilient. So this is based on the assumption that the complexity in your data is correlated or proportional to the complexity in your system. And so they use this complexity chain to then predict if your system is high or low resilience. They tested their model on a phytoplankton community, and the biological variables they inputted to the model include diversity, species abundance, cell density, biomass, chlorophyll A concentration, and then a function that's specific to phytoplankton. Two more approaches in this environmental systems category. Mean exit time or life expectancy was applied as a measure of resilience. And this approach was similar to the first one that looked at small perturbations over a time series, and they calculate mean exit time, which is the time it takes for a system to leave that stability basin. So also a similar idea of predicting the shift. And it's estimated from the fluctuations in the system. They applied the stability landscape framework and the Langevin approach, which is to calculate the fluctuations to then be able to calculate this mean exit time. They tested their model on cyanobacteria biomass over a time series in a freshwater lake. And this shows the abundance across the time. And then they calculated these two key components of the Langevin equation called drift and diffusion. And then using that, they calculated the mean exit time, which is shown with these green lines. And the idea here is that a shorter mean exit time suggests that the system shifts between states more frequently, implying that the system is less resilient. The next approach uses artificial neural network or a machine learning technique to predict water quality resilience. And they use this method to rank water basins based on their level of resilience. And the goal here is to be able to identify which water basins need what prompt restoration strategies. One of the limitations here is that the input they use for their model is this predefined validated index for water quality. And so if we were to apply something like this to the microbiome, we would need to select and define what kind of input we would use. So based on these approaches from using ecological models, this kind of makes me ask, like, can we adapt these methods or models to provide novel metrics for the microbiome? How can we adapt it? Is it feasible? Does it make sense? So the next category of approaches is in the area of human health. And these approaches look at the condition of the host or human and then infer microbiome health. So they don't necessarily measure microbiome directly, but the association is there. So this first approach identified a core microbiota, and based on the presence of that core microbiota or not, they inferred the resilience of children. They did a longitudinal study that characterized fecal microbiota of children in one daycare across 4 variables, including travel, antibiotic use, diarrheal events, and then age. And their goal was to identify which specific bacterial types might confer this increased resistance and resilience towards developing diarrhea. They define a core microbiota as bacterial species that are present in all samples for each individual child. And then this, oops, oh. Sorry. Okay, there we go. So this diagram represents just one of the analyses they did looking at a time series of bacteria composition from one child who had diarrhea 60 days before. And so they look at the change in observed species richness, so the number of species, and you can see that increases over the 60 days after the diarrhea, so it shows some recovery. And then they also look at the diversity in the community, and that also recovers. But as a comparison, when they did this with a child who didn't have diarrhea, their species richness and diversity recovered after closer to 45 days, 40 to 45 days, versus this child took 60 days. So then they infer that, they infer the resilience based on this presence or absence of the core microbiota. This other approach assessed the resilience of the host based on the microbial composition changes after a stress exposure in a rat model of PTSD. So if you kind of follow this diagram with me, they had a SPF is the stress exposure they tested. So they collected fecal samples and urine before the exposure, and then they had their SPF stress group, and then their control group. They let them rest for a little, and then they did behavioral and anxiety tests. And the output of the behavioral test was an anxiety measure that's already been validated in psychiatry. Based on the level of this anxiety measure, they divided the stress-exposed rats into resilient or susceptible groups. And then they looked at what bacteria are in the resilient versus susceptible rats. They did find a difference, but again, they don't really measure resilience. They just infer what a microbiome associated with the resilient host looks like. In the last area, these approaches are specific to measuring or estimating resilience of the microbiome. So this one from Shah et al. Applied a stability landscape framework to predict post-perturbation states and to infer resilience. And they use this approach to model the recovery of the gut microbiome after an antibiotic treatment. So they only assume that the system can be in 2 states. So one is a healthy state, or one is a diseased dysbiotic state. And then as one of the parameters, they use phylogenetic diversity. So how many types of bacteria in the community. And you can see that here. So this y-axis represents diversity. So a system with high diversity is resilient. A system with lower diversity is in this alternate state and has lower resilience. And then their model, because they're only assuming 2 states, they model a perturbation as an impulse. So opposite from the small natural perturbations, but an acute impulse. And then the idea is that this system can go into this one state, which would be one model, or it could get transitioned into this other state. So the goal of this approach was to really simplify the complexity in this ecosystem. This next approach uses functional redundancy as a resilience indicator of the human gut microbiota. And this approach created a network that links microbes to the genes in their genomes called Genomic Content Network. And this was to investigate the origin of functional redundancy. They applied their network and analyzed publicly available data sets of fecal microbiome transplant. And FMT could be seen as an acute disturbance. So it's when you take a healthy person's feces and apply it to somebody who needs a reorganized gut microbiome if they have like diarrhea or something. So it's a treatment method. So as to kind of explain what they did, this top panel shows a taxonomic profile, so what bacteria are in the samples. And then they created their genomic content networks. And this is just hypothetical to show the conceptual model. So you link the taxa to a gene, and this left one represents each taxa has a unique genome. So the functional redundancy in this community is very low. This one represents some taxa have unique genes, but then there are others that share the same gene. So for instance, this purple taxa shares the same gene with this red taxa. And then this very right graph represents a highly functionally redundant community because all these taxa share similar genes from the similar genomes. And then they calculate functional profiles of each sample based on functional diversity, which is how many functions are within a sample, and combine that with functional redundancy to calculate that profile. And so this left plot kind of represents a community that has high functional diversity, but low functional redundancy. And then this middle one, it's comparable. And then this right one shows that although they have low functional diversity, so there's not that many functions occurring in this microbial community, but because the genes are redundant, they have high functional redundancy. So then they would term this as a resilient microbiome. And when they did the analysis with the fecal microbiome transplant dataset, they found that resilient recipients of the treatment who have a high resilience or high functional redundancy, the treatment did not work as well because their microbiome was so stable. The donor microbiome didn't stick. And then the last approach, they predict the metabolic potential and use that as a resilience measure for gut metagenomes. So they use shotgun metagenomics, and they estimated resilience through bacterial richness, diversity, and evenness specific to this microbial byproduct called short-chain fatty acids, or SCFAs. And it's well known that these microbial molecules or products help regulate our gut and maintain health. And the three main ones are propionate, butyrate, and acetate. So they use these to infer resilience. And they tested their approach on shotgun metagenomic data sets of 12 populations. And they compared individuals from an industrial setting to non-industrial. And they compared these two populations because it's known that individuals who are in industrial settings have a lower abundance of SCFAs. And then based on the gene counts, they predicted functional profiles. So one of the questions I got from this approach is, could we use SCFA counts or abundance as a potential metric for resilience for a microbial community? So then that brings us into what gaps are still present. The effect of the daily natural disturbances versus acute kind of impulse perturbations, the difference in those effects are still not very known on how they affect gut microbiome resilience and stability. the methods for quantifying gut microbiome resilience has not been established, and the model or guidelines for assessing resiliency and how to best correlate them with clinical outcomes is missing. And then best practices for quantifying resilience of the human gut has not been provided yet. Some of the opportunities that are available in this area, assessing the feasibility of applying these different ecological methods and models to measure gut microbiome resilience is still needed and could be done. So for instance, assessing how to apply artificial neural network to microbiome metrics or the mean exit time to best measure resilience of the gut microbiome, assessing that kind of models would be an opportunity. Comparing those methods and evaluating metrics in relation to host health, and then further defining assumptions, and then also identifying what kind of data would be required and challenges and limitations for applying those methods and metrics. The next level of opportunities that are available would be to develop a framework that's reproducible for measuring gut microbiome resilience. And then in this area, we would need to evaluate the definitions and metrics and characteristics as well. Kind of down the line, or ultimately, one of the opportunities would be to integrate gut microbiome resilience data with host clinical outcomes. To develop the most biologically accurate measure of overall host health, we would need to consider what kind of data we need to look at, and looking at time series data and doing longitudinal analysis to compare the changes across time to be able to capture the fluctuations would be crucial. Considering what kind of disturbance I model is also crucial. So do you model impulse disturbances or the more small and natural daily perturbations? Defining what a baseline state looks like and what a resilient response looks like is crucial too. And then what kind of host response would be defined as healthy? And that one would probably be challenging, but that's key. And the last consideration with applying resilience to gut microbiome as a metric is that resilience is very context dependent. So it's very context dependent. So based on the system, the characteristics of resilience, and the metrics and indices that are applied would differ. In conclusion, assessing the feasibility of gut microbiome resilience as a measure of host health would require kind of looking at surrogate measures, definitions, metrics, and considering what kind of data types are needed and the availability of data. A metric for overall health, especially using gut microbiome as a proxy, applied in research and translation, would help us understand the relationship between health, our health, human health, and gut microbiome stability. It also help us kind of better understand what the drivers and mechanisms of chronic inflammation. We could also improve treatments and diagnoses and prognoses, predict disease severity or risk better. And also with a metric like that, we could monitor lifestyle changes, disease progression, or treatment. Okay, and thank you for listening. I'd like to thank my advisor, Shannon, and all the DMICE faculty and administration, and also my DMICE students and fellows. Thanks to the grant.
Great, that's fantastic. So we're now going to have time for questions, and there's one that goes back to the twin. So I guess they're asking for a clarification, Christina, which is asking if the twins are identical.
Yeah, I'd say so. Yeah, I was assuming they're identical, just as an example to show, you know, two individuals that can be as similar as possible.
Right, because then you'd really be focusing, you'd be able to remove the genetic component and just think about environmental.
Mm-hmm.
Right. Yeah.
Yeah.
Okay. Other questions for Christina? And you can obviously turn on your unmute and ask a question directly, or you can put it in the chat, either one.
Hi, Christina. This is Mike Mooney. Great talk. I was wondering, you talked a little bit about longitudinal analysis. I just want good data on the time scale at which, if you had a magic pill to correct your microbiome, how quickly does that translate into changing health state?
Yeah, that's a very good question. There's one study, I'm trying to find it. I can't find it. But there is one study that suggested 12 time points is kind of the key to accurately capturing like changes in the fluctuations in the system. But I didn't really see any study that assessed that for microbiome changes specifically. A lot of the studies, let me go back to it. So for instance, like this study that looked at antibiotic treatment, they used a data set that was like a year long. And then this one that looked at the diarrhea in the children, they also looked at a data set that was a year long. So,
Okay.
is that what you're asking?
Yeah. Thanks.
Any other questions for Christina? Any ideas? Remember, this is her symposium. So you have the ability not just to ask questions about the talk, but just to kind of think about what she's laid out in terms of her ideas. And obviously, you can always email her directly if you'd prefer not to do it this way. But we just love to get your input for her. This is true for all of our students with their symposium. Any ideas you have, or if this resonates at all.
Hey, Christina, this is Sean Gibbons here. Great talk. I had a kind of devil's advocate question for you. So resilience is one way to measure sort of maybe like a health state for the microbiome, but you could imagine disease states are resilient as well, right? I could maybe argue that recurrent C. diff is a resilient state. Have you thought about that?
Could you explain the second part about disease state resilience?
That potentially dysbiotic or suboptimal
Mm.
ecological compositions of the microbiota can themselves be quite resilient and
Yeah.
have measures of resilience. And so
Yeah.
resilience is sort of almost an agnostic, not has no good or bad flavor
Yeah.
to it in that case. It could be either or depending on context, right?
Yep. Yeah. I think that's a really good point. And I'm kind of holding myself back from getting too far into this because it gets into proposal stuff. But my thinking is it's kind of crucial to include host health information to associate with the state conditions, so the resilience. So then, based on what the health outcome is, whether it be chronic inflammation or dysbiotic, we can associate that with the resilience. Yeah, but that's a good question. I haven't really thought about it too much. So I think having that component of the condition of the state would be key. And then we could work on how to best kind of define that and a measure that would capture it. If that makes sense, but that's a very good question.
Great, thanks. Thanks so much.
Just as a note about that before we go to the question, just kind of adding on to what Sean said, this would be really relevant in cancer, right? Where we think about the evolution and kind of ecological aspects when we think about the tumor, especially thinking about resistance, et cetera. So resiliency of the cancer as well, right? Would be a different interpretation. Okay, so your next question is from Chris. He said, what would a long-term vision of the clinical application of a better understanding of the gut microbiome look like? Would this be more proactive health recommendations or monitoring acute treatments or both?
So if I'm understanding your question right, Chris, there are many proposed like applications of something like this. So one of you mentioned like proactive health recommendations, using a metric like this to kind of monitor how somebody's lifestyle changes improves their health. So kind of more for monitoring improvement, which could also be applied to treatments and to monitor how well a treatment is working or how it's not. So it could be applied in that area too.
And I think the question is for you, right? It's kind of what you, I mean, anyone could answer, but if, you know, if we talk about a long-term vision, right? So we think about the significance of this work if it was successful, right? So what is the future of clinical applications in your opinion, right? If you had a, you know, you could say if you had a crystal ball, but you could also just base it on what you're thinking. So is the idea of more proactive? Is it the monitoring an actual, you know, therapeutic aspect, or both, or something different?
I guess I was thinking both, but I guess I was leaning more towards using it as kind of a predictive measure to predict, for instance, in clinical depression, this concept of resilience has been applied, and it's found that individuals with depression kind of show this critical threshold or tipping point into a state that's associated with clinical depression and symptoms. So if we could use this metric to kind of predict if somebody's getting closer to that tipping point, so then we can apply, I don't know, whether it be medication or suggest more exercise or a healthier diet to help that person stay in the stable state. So it would be a measure to predict somebody's health to avoid a shift into that unhealthy state. That's kind of the main application that I'm interested in.
Chris, do you want to add anything to that, or does that answer your question?
Yeah, that does answer my question. I guess, you know, the first thing that comes to mind for me is things like dietary recommendations, because that's, you know, pretty low cost and practical. But you know, the monitoring stuff's interesting. It just seems to me that for um, you know, to like predict a change in someone's treatment, I'm just curious of like how practical that is to implement, because, you know, if you have to take like, is that like regular fecal samples or something like that? Like, you know, it's like patient compliance. You know, it seems like that could potentially be a challenge if it's more of a hassle. I don't know. I'm just curious like what the implementation would look like. But
Yeah.
it seems like there are a lot of possibilities.
Yeah, there definitely is. And I think one of the advantages of finding a measure using the gut microbiome to estimate that overall health is that you can just look at the fecal microbiome or a fecal sample. And so that's not very intrusive. You know, the frequency at which you have to sample is unknown. So if it's like every day, that wouldn't be very practical. But for instance, if it was like every 3 months, maybe that's a little more practical for a patient to go in or provide a fecal sample, and then you can do simple 16S to look at the diversity of the bacteria to kind of assess if they're improving or if they're not progressing as much as they want. So yeah, I was thinking about fecal samples.
Yeah, that makes sense. Thanks.
Great. Any other questions for Christina? She did put her email in the chat if you really want to follow up with her. But anyone else? Okay, well then we'll just join me in congratulating her again on a successful symposium. Great job, Christina. Okay. Thank you all.
Thank you.
Ready?
Okay. All right.